And you can tell it's going because I'll have this little circle here at the top. Just letting you know. Okay. All right, so now we'll start. Okay, so, all right. There's another good reason to always take notes in class. So basically, this ubiquitin protein ligase is the component that's going to determine the specificity. So that's what you want to focus on to determine how the degradation of something's regulated. Because this is what's actually binding the substrate, and it determines when it's going to bind the substrate. Uh-oh. Okay. All right, so the ubiquitin protein ligase that we're going to be dealing with here for this transition are these colon ring ubiquitin ligases. So I had the first paper on this whole gene family, these colons. It was like 96. So, because my lab actually studies these things. But this particular kind is called SCF for skip1, F-box, and then col1, SCF. Okay, so the way it works is this. This col1's a scaffold. It's got an adapter. It has different substrate receptors. This binds the substrate. It's got a ring finger protein that binds the E2. So just to remind you, we're talking that whole complex. Like here it's drawn like this E3 is just one thing, but the E3 can be a multi-subunit complex like that. And it's going to be interacting with the E2 ubiquitin conjugate enzyme here. So it's going to be binding the substrate, and it's going to be binding the ubiquitin conjugate enzyme, allowing the ubiquitin conjugate enzyme to add on the ubiquitin. Okay, so here we have this part is the E3, ubiquitin protein ligase. It's going to bring in the ubiquitin conjugate enzyme, the E2. So it's going to bind the substrate, and in this case, it's going to be regulated by phosphorylation. So when the substrate gets phosphorylated, it's going to bind it. And then the one E2 will come in with the activated ubiquitin and transfer. Ubiquitin, remember, is just a small protein, 76 amino acids. Another one will come in and add it to that ubiquitin. Another one will come in and add it to that ubiquitin. When you get 4 ubiquitins of the right linkage to each other, then that substrate gets degraded, and then it's going to get degraded by the proteasome. Okay, so this is how you actually regulate the G1 to S phase transition. Remember, we're focused on how are you going to make this CDK S phase cyclin complex active, and it's inactive because it's bound to the CDK inhibitor. So this complex, this SCF complex, is going to recognize the SIC1 and degrade it, but it's only going to do that when the SIC1 is phosphorylated actually on multiple sites. Okay, so it has to be like 7 phosphorylations. That phosphorylation is going to be done by these G1 cyclin CDK complex. They're going to phosphorylate SIC1, but they're phosphorylating it on 7 sites. So the way this thing works is this phosphorylation is unstable because there are phosphatases that can take it off. So the CLN can phosphorylate, and then the phosphatase takes it off. And then it can phosphorylate and takes it off. Maybe it gets 2 phosphorylation sites, and then it takes them off. Maybe then 3, and it takes it off. Hey, can you go outside and put a door in front of that?
What do you?
Can you go outside and put a door in front of it? I don't know how you can put a door in front of it if you're like inside. You know, and close it and have a door behind it. Go outside with a door, with a chair, put a thing on it, and then walk around back in. I think you've already heard this lecture before multiple times. Okay, great. It's good. She doesn't need to know this. All right. Okay. So here we go again. So what's happening is that this G1 cyclin CDK complex phosphorylating it, but it's being undone by the phosphatases that come off and do it. So you're going to have to accumulate a lot of this to be able to hit it on 7 sites before the phosphatases can take them all away. Okay, and that's going to allow you to have enough time to stay in G1 phase. Now these things are also phosphorylating themselves on that PES domain, and then this complex is degrading them also. Okay, so it'll take a long time to accumulate enough of these because they're also being degraded. Okay, and so that's what allows G1 to last that long, because because while these are being degraded, you have to make new ones, and it's going to take a while to make enough of them that there's now enough CDK1, G1 cyclin to phosphorylate this on 7 sites before this thing can hit it, okay? But now, like I told you, these transitions are super fast. And the way that this one works, okay, you got a positive feedback loop, and the positive feedback loop is this. When this thing gets degraded, it's going to produce S-phase CDK complex. The S-phase CDK complex is also able to phosphorylate SYK1. And then now you've got these guys phosphorylating SYK1, and now the one that's released phosphorylating SYK1. And then when this gets degraded, it produces more of them, which now can phosphorylate it again. You see what I'm saying? So once you start making this S-phase cyclin CDK complex active, because it can also phosphorylate SYK1, you're going to rapidly activate all of them. Okay, so it's very fast. The cell wants to be in G1, and then it wants to be in S-phase, and it doesn't want to like take its time doing it. It wants to go very quickly through this positive feedback loop where the S-phase cyclin also is involved in its own activation. So any questions about this? Okay. All right, so it turns out that this is in fact the only, this substrate, SYK1, is the only essential non-redundant substrate of the CLNs. Okay, so what does that mean? If you get rid of the CLNs, remember you don't go into S phase at all, right? You're stuck in G1, they die. I mean, they grow and then they die at some point. But if you get rid of the CLNs and you get rid of SYK1, okay, so now you got rid of both of them, got rid of this, you need it to go to G1 to S phase. But now you get rid of this, which is its target. Well, the cell will go into S phase, okay? So it won't die. So that tells you that you don't need the CLNs if SYK1 is missing, okay? SYK1 is the only substrate that these guys have to, it's the only essential substrate. And by that means that if you get rid of these, the yeast is dead. But if you get rid of these and this, now the animal's alive. So that makes it an essential substrate. It's essential for life, for viability. Okay? So, and then, the only problem is that they're a little sick because they go into S phase too soon, before they're ready. So they haven't prepared everything for S phase. So when you don't have this, they'll go in like earlier into S phase. Okay, so this is the and by non-redundant, it just means that no other kinase can hit SYK1, because if some other kinase could hit it, then if you get rid of this, well, the other kinase X would hit it, but there is no kinase X that can hit it. If you get rid of these, it doesn't get phosphorylated, and you never get into S phase. Okay, so it's like a genetic argument. And that genetic argument is listed out here, and it's basically that you need to have, if you don't get rid of all 3 CLNs, you get a G1 arrest. But if you get rid of the 3 CLNs and SYK1, its target, then the yeast are viable. Okay. Oh, let me switch this thing to HD. Okay, that's right. All right, so now we're going to talk about, so that was yeast. Now we're going to talk about the mammalian G1 to S phase transition. All right, and this gets started on RB tumor suppressor protein, different protein we haven't talked about yet. I'll go into this. I'll go into a little bit more about how this was figured out when we talk about cancer later. But basically, this is a cross section of the eye, and the retina is the back section of the eye that contains the photoreceptors. So it's actually got so the light's going to go through the cornea. And, it's sort of interesting. This is sort of cool in a way. The cornea is a thick layer of cells. So when you're seeing things, the light's going through a thick layer of cells. Then it's going to go through here. It's going to go through this lens. It's going to go through this open space here, okay? And then it's going to go through this layer of cells, which are neurons, okay, that are connected to these cells, and it's going to go through the cell body here, and then finally hit the receptor. So we're going through a thick layer of cells here. We're going through, and then through all of these cells here before it actually hits the photoreceptor part that can take up the thing. So it's sort of amazing that you can actually see that well because you're actually the light's going through multiple layers of cells in your eye. At any rate, that really had nothing to do with this lecture, just sort of interesting. The RB, if you don't have the RB gene, then if you knock out both copies, then you'll get retinoblastoma, which is a tumor of the retina. And we'll get into the genetics of this when we go into the cancer lecture. But basically, this is one of the few examples where a single gene knockout will cause cancer. Generally, for solid tumors, you have to have multiple hits on different genes. But here, this one gene will cause cancer. And so it's a childhood cancer where they end up with multiple tumors because they've already got one copy of the RB gene knocked out. And if they get another copy knocked out, then wherever that gene is knocked out, they'll get tumors. They'll get multiple tumors back here. Okay? So that was the first tumor suppressor gene to be cloned. All right, and we'll get into the two-hit hypothesis later. Right now, we're just going to go into what is RB doing because it's involved in this G1 to S phase transition. So in mammalian cells, the decision point for whether or not to go from G1 to S phase is in G1 phase. It's called the restriction point. And so once the cell goes through this, it's going to go through the cell cycle. So if it gets through this restriction point, it'll always go through the cell cycle. And we're going to be looking at what RB's doing here. Okay? So RB's job is it's an inhibitor of a transcription factor called E2F. Okay? And E2F, you know, as you probably learned in these transcription factor lectures, they generally work as dimers. And it works as a heterodimer with another transcription factor called DP. And it's going to turn on genes required for S phase. Okay? And if you overexpress E2F, you're going to get into S phase sooner. And if you inactivate E2F, you'll never get an S phase. And if you reduce it, you'll go in later. So this transcription of S phase genes is sort of the trigger, determines when you get into S phase. But as long as RB is inhibiting E2F, they're not going to go into S phase. And so what happens is here we have the mammalian cyclin D, which is the G1 cyclin. And in mammalian cells, you have different CDKs for different parts of the cell cycle. So CDK4 or CDK6 is G1 CDK. This complex, its main substrate is RB. It's going to phosphorylate RB, and that's going to make RB release E2F, the transcription factor. E2F now binds its binding partner, turns on S phase genes. Okay, so that's how it works. And you can there's like a genetic argument that you can make to say that this is the only essential substrate here also. So it only has one major substrate. P16 is one of these CDK inhibitors. And so if you overexpress this, it's going to block the CDK cyclin activity, and then it can't phosphorylate RB. So RB is always going to be bound to E2F, and the cells are not going to go into S phase. So then you know, if you then don't have RB, now you overexpress P16, so you inactivate the G1 cyclin CDK complex, nothing happens, okay? The cells go into S phase easy, all right? So what it's saying is that the only essential substrate is RB. And you can get those cells that lack RB because every time you make, like if you make a cell line from the RB, the retinoblastoma knockout, like, you know, there's cancer. You can just take those cancer cells and start growing them. Well, I mean, they lack RB, right? So they don't have this checkpoint. They're going to go into S phase regardless of whether they're supposed to or not. And once they make that decision, they're going to get another round. So that increases that. It's not that cancer cells divide too fast or anything. It's just that they keep dividing, right? And so here they're not any kind of regulation say, hey, you know, we made a nice retina, stop dividing. All that regulation's gone because it's going to just keep going through the cell cycle. Okay, can't respond to that restriction point. All right, but I will say that that this is a little bit simplistic. Okay, and it's a little simplistic because there's actually multiple E2Fs in mammalian cells. There's positive E2Fs, which turn on S phase genes. But there's also repressing E2Fs. And these repressing E2Fs are also bound to DP, and they're also bound to RB. okay? But in this case, they're repressing E2Fs, and they're also able to bind DNA, okay, because of transcription factor. So they're active in binding it, but because they're repressing E2Fs, when they bring in RB to this region, it represses the transcription, okay? And they're going to be inhibiting the transcription in part because RB can interact with HDAC, histone deacetylase. And just to remind you what that is, so if you remember from when your chromatin lecture or your transcription lecture, the histone acetyltransferase is adding on acetyl groups onto the arms of the histone tails, and then the histone deacetylase is removing it, and then you guys learn why those histone arms are like. What's the answer? Why does it? How is it making any difference whether or not the histone arms are acetylated or not? What's that? Raise your hand when you answer. I can't tell who's talking. No, I inhibited you. What's that? Well, I mean, so the question is, what is, why are these arms, like how is acetylation affecting these arms versus these arms, and why is this a closed conformation and this open conformation? What's happening? And how does that work? Well, it's not unwinding the DNA. You got to raise your hand. So what is it? Yeah, but why?
So the DNA is negatively charged, and the histone tails would be negatively charged. They repel.
Yeah. I mean, that is correct. Except acetylation doesn't actually add a negative charge. Acetylation just gets rid of the positive charge. So, but that's basically correct. So it's if you look, I mean, I have that slide, but then I was like, well, you guys already had it, so why put it in? But basically, what happens is you got the DNA around that histone loop. It's negatively charged. These arms are like this, okay? But if they have positive charges, they're going to be on the side of the DNA on that histone thing, locking it in place. Okay, it's not going anywhere. You have to have them loose, and now the DNA is sort of loose. It can be accessed, right? All right. So now we're going to be talking about the importance of the cyclins in mammals. So this is that diagram of the different type of mammalian cyclins. You've got your G1 cyclin, cyclin D, and D1 and D2, D3, and so this is generally consider your G1 cyclins. This is your G1S cyclin, cyclin E. You got S-phase cyclin, cyclin A, and mitotic cyclin, cyclin B. And, they all have different roles, and you think they might be important for different parts of the cell cycle. Like, this might be important for G1, and this one might be G1S transition, like that, right? So here's an experiment. This is a tissue culture experiment where they're growing the cells on a plate, and they're going to ask, well, how important are the G1 cyclin Ds? All right. So what they're going to do is they're going to have the cells in G0. Remember, that's that quiescent phase. You take away growth factor, which is receptor tyrosine kinases, they're going to go out of the cell cycle into a G0. And then you add back the growth factor. They're going to come back in the G1 phase. And during that time, you're going to add BRDU, which is that thymidine analog that tells you whether or not, like if it's going to turn into like, instead of a DTTP, it'll turn into a BRDU TP or whatever. It'll get put in wherever a thymidine base would get put in. And so a T base, instead of a T base, it'll be BRDU. So you can follow it, okay? You can follow DNA replication by asking whether BRDU got put into the DNA. All right, and so you're going to have two sets of cells. Both of them are coming back into the cell cycle, and this one you've injected an antibody against cyclin D. So the antibody is going to bind cyclin D and keep it from working. And so this time 0, you add the growth factor, put them back in the cell cycle, and then this is the time when you're adding the antibody to block it, and then this is the control. and you're saying, well, how much DNA replication was there, okay, at 16 hours? So, here, if you add the antibody against cyclin D early, before they enter S phase, you don't get any replication. And then if you do it at 12 hours, you don't get hardly any replication. But if you add it later, when they've actually already entered S phase, it doesn't really have much effect. Now they'll incorporate DNA, all right? So these G1 cyclins, according to this experiment, are important to get into S phase, but they're not important for S phase because you can actually do the labeling after they've already started, and it's fine as long as they're already in S phase. And this is what the actual experiment looks like. Here we're looking at the nuclei of 4 cells, and then here's where they've injected their cyclin D antibody into these 2 cells, so it's red. And then if you look at where BRDU was incorporated in the nuclei, you see that the 2 cells that were not injected have incorporated BRDU, that means they're in S phase, and the 2 cells that did get the antibody did not incorporate BRDU. Okay, you don't see it in the nucleus. That's just diffuse staining in the cytoplasm. All right, so what this experiment's telling you is that cyclin D seems to be essential to get into S phase, or at least it delays it like crazy. Okay? So then, these knockout mice people, they like to knock out genes and then say what's happening in the normal situation. So they're not satisfied with that tissue culture experiment. They're like, okay, I'm going to knock it out and see what happens in the actual mouse. So now they're going to knock out, and so because there's 3 of these cyclin Ds, D1, D2, and D3, they're going to knock out all 3 of them. Okay? They're going to knock it out and say what happens to the mouse. So let's take a look at what happened. So here's a control mouse at E15.5. Okay, so E15.5 just means 15.5 days after they mated, okay, after fertilization. And you can see that these animals here that have locked cyclin D1, D2, and D3, they're going to arrest at E16.5, so a little bit later than this. Okay, they're going to arrest and they're going to die. So what would be your conclusion? What would be your conclusion about that? Well, you would say it's important for life, right? But, if you were trying to say, well, is it important for cell division? Like, if you see this little mouse here, would that be important? Can you say that cyclin D is important for cell division? No, because there's like hundreds of millions of cells here. Hundreds of millions of cells were made with no cyclin D. In fact, they formed eyes, they got little hands, they got like a circulatory system, a little heart. I mean, that's like a ton of development, and they didn't have any G1 cyclins. So that initial experiment where they were injecting tissue culture cells, sort of misleading, because when you actually knock it out in the mouse, not so important. I mean, it's important. You can't live, right? They can't live if every embryo dies at E16.5, but in terms of cell division, not that important, okay? So then they said, okay, so well, what if we instead of knocking out the cyclin Ds, we knock out the CDKs, the G1 CDKs that bind the cyclin Ds, so they knock out CDK4 and CDK6, and then they get the same thing, all right? So it doesn't matter whether you knock out the G1 cyclins or the G1 CDKs, you're going to get the same effect. Millions of cells are being made. All right, so then they knock out CDK2. Now this is the G1S cyclin, okay? So it's bound to cyclin E. It's a different cyclin, and it's important for the G1 to S phase transition. And then this one's even worse. This one produces a mouse that can become adult. So you can actually have an entire mouse growing up, and it's healthy, it's running around, it's eating, it's interacting with its neighbors. It doesn't have G1S cyclin, activity at all. But it does have meiotic defects, so it can't have any children. And then if you knock out the G1S and the G1 CDK4, but again, you've only knocked out one of the G1s here, and then it's 15.5, but you still have the CDK6. Remember, you have CDK2 and CDK4 and CDK6. CDK4 and 6 are G1, so you still have that one, right? So it's not surprising that that's only slightly worse than this. Okay, but here they said, okay, let's get rid of all of the S phase, all of the CDK4 and CDK6. So we're getting rid of all the G1 and the S phase CDKs. Okay, so that's CDK2 for G1S, CDK4 and CDK6 for G1. Okay, so they're going to knock out all three of them. So they're making a mouse, and it turns out that if you just have CDK2, one copy of CDK2, then then you're actually okay. You can get past that initial point, which in this case is 12.5. All right. So here you can get past 12.5 in this animal that has one copy of CDK2, but then when you get rid of all of them, they arrest at 12.5. And now, what's your conclusion now? So we got rid of the G1S and the G1 CDKs. So what's your conclusion about whether is it required for cell division? Huh? No, it's not. Now instead of hundreds of, so instead of maybe like 700 million cells, maybe we only have 100 million cells. Doesn't change the fact that there's millions of cells here. If you look at this carefully, you can see that they made the spinal cord, they got an eye. I mean, they're basically, sure, it's earlier arrest, but you still got millions and millions of cells being made when you knock out all of these CDKs. Okay, so that's telling you that even the G1 and the S phase CDKs not that important for just basically dividing your cells. Okay, so then they say, okay, let's knock out CDK1. So CDK1 is the mitotic cyclin. And if they just knock out CDK1 alone, they don't actually get any embryos. And generally when they don't get any embryos, they'll go into the mother mouse and they'll chop it up into really fine pieces, and then they'll start looking for it. Okay? They'll put them on slides, and they'll cut up the uterus, and they'll look for the little embryo that's like, because like these embryos, they were going to die in the mother, and so they cut the mother open, and then they saw, where they arrested, right? So here they're looking, okay, where's the CDK1 embryo? So they're taking the mother, and they're chopping it up, and they're putting them on slides, and they can't find it anywhere. It's like nowhere. So they can't even see anything. So what they think is that they didn't even get a single cell division out of CDK1. And yet, when you get rid of the CDK2, which is G1S, or CDK4 and 6, S phase, you still get all these millions of cells. So what it turns out, the way the story unfolds is CDK1 can, even though it's not even normally expressed at this point, can get expressed earlier, and it can actually bind to the G1 cyclins and the G1S cyclins to make a complex that it normally never makes, okay? So CDK1, the mitotic cyclin, can now bind to G1 cyclins and G1S cyclins, and it can phosphorylate RB, it can like liberate E2F, it can do all those things that it's normally supposed to do, okay? So CDK1 by itself can do the whole thing if it has to. I mean, you can't make a viable mouse, but you can get, millions of cell divisions out of that, okay? And then just keep in mind that CDK1 is the mitotic cyclin, but it's also the cyclin that's most closely related to the CDK1 in yeast. And in yeast, there's only one CDK. CDK1 does everything, okay? It does all the stages of cell cycle. And so the fact that the mitotic cyclin can still do all the phases of the cell cycle just says that part has been conserved all the way from yeast to mammals. Okay. All right, so now we're going to talk just really briefly about checkpoints. So checkpoints are something bad happens to the cell, and the cell's going to stop the cell cycle, fix it, and then go on. So there are different checkpoints. We'll talk about some other ones later.
Before you said cyclin, you get into S phase, right?
Well, I said that tissue culture cells experiments suggested that. But then when you actually knock it out in mouse, yeah, it's not. Yeah. I mean, and you got to take these with a grain of salt anyway, because I mean they're just looking over a period of 16 hours, right? So it's like it's. And the other thing is that like if these things were never made, then you could maybe get a compensation increase in CDK1 expression, So the cell, the mouse can say, whoa, I don't have any cyclin D. Let me upregulate these other genes to compensate for it, right? All right, so we're talking checkpoints. So basically, when something bad happens to the cell's going to say, oh, let's stop the cell cycle and let's fix the problem. So one problem it's going to want to definitely fix is DNA damage. Because if you're going to replicate in DNA damage, you can fix the DNA damage in there, like if you put it into the 2 copies, and it's there forever, right? So the cell wants to, if it gets DNA damage, it's going to want to stop the cell cycle. Okay, and the way it's going to do that is through P53, and this is also a tumor suppressor protein. This is actually the most commonly mutated in solid cancers. About 80% of all solid cancers have mutations that get rid of P53. Okay, so it's very hard to get a solid tumor without mutating P53. Okay, but here we're going to talk about its normal job, and its normal job is to take DNA damage signals, and it's going to stop the cell cycle. All right, so this is the way it works. Okay, so P53 is a transcription factor, and when it's made, it's degraded by MDM2, which is an E3. So remember, the E3 is the ubiquitin ligase, ubiquitin proteolytic pathway component that binds a substrate. So MDM2 is going to bind P53. It's going to cause it to get polyubiquitinated. You add ubiquitin to it, get degraded. Okay. And then another P53 is being made. And MDM2 is going to cause that one to get degraded. Another P53 is being made. And then MDM2 is going to cause that one to get degraded. And these are just inhibitory symbols. Just means that it's inhibiting, that thing. In this case, by degrading it. So P53 doesn't have any chance to accumulate. Okay, why is that important? Because P53 works as a tetramer. Okay, it works for them together. It can't work by itself. Okay, but you're never going to accumulate enough because as soon as you make it, it gets degraded by MDM2. Okay, so as soon as there's DNA damage, and the DNA damage checkpoints is usually done by like a checkpoint being here, and we're not going to get into the pathway for this class, but basically it's ATM, ATR, kinases get activated here. They're going to activate another kinase called CHEK1, and they're going to end up phosphorylating P53. So this part is going to be important, okay? So a kinase, like this part's not important. Okay? But this part is important. It's phosphorylating P53. Suddenly, when P53 is phosphorylated, MDM2 can't bind it anymore, okay? So now you're going to start accumulating P53. It's phosphorylated. Okay? Now you start accumulating enough P53. P53 is going to form a tetramer. So the tetramer is a transcription factor. Okay? So now the tetramer, because it's no longer being degraded, is going to turn on gene expression. All right, so here it's made a tetramer because there's DNA damage, and now it turns on transcription, and it's going to turn on a lot of genes that are going to help to repair the damage, like a lot of repair proteins. But it's also going to stop the cell cycle, and the way it's going to stop the cell cycle is it's going to make a CDK inhibitor. Remember, this is the protein that binds the CDK cycle and inhibits it, called P21, and that's going to bind CDKs and inhibit it, okay? Now this diagram is from an old textbook, and I like it because it shows the tetramer, but this part is not correct. So you guys remember PCNA? PCNA? Oh, you guys didn't do DNA replication. No DNA replication this year. Yeah, and I don't think you even really go over that much in genetics. Anyway, All right, so I'll give you a quick overview on pCNA. So pCNA, okay, so here's your DNA, and you've got a polymerase. Let's say DNA polymerase. All right, so what pCNA is, it's this big ring clamp like this. Okay? It's got 3 components in thing, pCNA. So what happens is the DNA polymerase will go on the DNA, it's going to fall off. And it'll come back on and go on the DNA and fall off, and go on the DNA and fall off, and go on the DNA and falls off. It doesn't have much processivity. But what will happen is once you load it on, the DNA polymerase plus pCNA bound to the backside of DNA polymerase. Now the polymerase wants to fall off, but it can't fall off because the pCNA is on the DNA. It's around the DNA. It's like a ring around the DNA. The pCNA can't get out of the DNA because it's around the DNA. So no matter what polymerase does, it can't leave. Okay, so it increases the processivity. It's part of the DNA replication complex. So anytime you have a replication fork, you're going to load pCNA there. Okay. All right, so they found that P21 is bound to pCNA. And they're like, whoa, that's going to inhibit DNA replication too. So it's actually working 2 ways. Okay, but watch, don't write that down. It's not true. It doesn't. And the first clue was there's 10 times more pCNA than P21 in the cell, even under these DNA replications. So there's no way that it could inhibit all the pCNA. There's 10 times more pCNA. And then my lab and 2 other labs showed that when P21 binds to pCNA, now it's degraded. So it's not that P21 is inhibiting pCNA, it's that this is a way to get rid of the P21 that you induced when the DNA damage, okay? Because when there's DNA damage, the pCNA is now on the DNA and it gets just degraded. So it's just a way of degrading the P21 that's made. Okay, so just ignore this part of the pathway. You don't need to know it. I just told you stuff that you didn't need to know. Okay? This is the old slide. All right. Okay, so there's another, so we'll get into other checkpoints later, like in mitosis, but here's another protein, a CDK inhibitor that is related to P21. It's like, it's what's called a paralog. It's similar, but it's a different gene. So this gene is involved in, let me go here to the overhead. Here's this normal cell cycle we saw before. I thought you fixed that thing, Snail.
Nice.
Maybe you need to lean it against it.
Maybe I'll just stand out there and hold it.
No, don't do that. I might need something.
I'll put another chair.
Yeah, that's fine. Let's do multiple.
You don't want me to stand out there because you might need me.
Yeah, you might need to get me something or who knows what. All right, so here's the cell cycle. And then what happens is when the cell does what it's supposed to do, like, you're talking to an organism. And let's say you've done all the divisions you need to make that particular organ, then you're going to leave the cell cycle and you're going to go into G0, okay? And then you only go to G0 back again when you get those, those growth factors, which, as I told you, are receptor tyrosine kinase ligands, generally. Okay, but at any rate, when let's say you're making a kidney, okay, and the cells have made enough kidney, and they know they made the kidney the right size, they're going to leave the cell cycle and go into G0, okay, and they're going to wait. And if you damage your kidney, they can now fill it up again. But until then, they've left the cell cycle, right? So what happens is that this P27, it's a CDK inhibitor. This thing is important to go to G0. If you knock it out, the cell won't go into G0 when it's supposed to, and it'll go around 1 or 2 more times. Now, it doesn't go around forever. It doesn't form a tumor. Eventually, something else will stop it. But all of the cells in that mouse that is lacking P27, you'll get more. So you'll have more kidney, more, almost everything, more thyroid, more skin, more everything. So what happens is you end up with a bigger mouse. So everything is still sort of normal. The mouse is fine. In fact, it can have children. It's just bigger. And it's bigger because it has more cells because the going to G0 at the right time during development didn't occur. All right. So that's it for that one. Now we're going to go to cell cycle 2. So we're like way behind. But on the other hand, we've got lots of time to catch up on the last lecture, hopefully. All right. So I had to throw this in because nobody's talking about DNA replication before me. So we're not going to go into DNA replication per se, like the actual polymerase is involved and pCNA. We're just going to go into how the initiation of DNA replication is regulated and what keeps you from doing it too much of it. Okay, so here we're again looking at BRDU labeling, and if you label cells early in S phase with BRDU between 0 and 2 hours, and then you wait till they go on mitosis, and then you get a chromosomes condense, and then you ask where on the chromosome is the BRDU incorporated early in S phase. Okay, so you're labeling S phase, and then when the cell goes through mitosis, you're getting the chromosome and asking where is it. Okay, in between, you've washed out the BRDU. Okay, so and then you can do the same thing for middle S phase, 3 to 5 hours, or late S phase, 6 to 8 hours, and this is all one chromosome. Like I don't know which chromosome it is. Maybe it's chromosome 1. Could be chromosome 13. At any rate, what you see is that when you look at that chromosome, certain regions will replicate early in S phase, some will always replicate middle S phase, some always replicate late S phase. Okay? And so that means that when the DNA replication starts, it's sort of regulated. Now, one correlation is these late ones are generally heterochromatin, the condensed DNA, and these are generally more open areas. But that's just a rough correlation. So the initiation in a normal cell cycle is regulated. They're going to start at certain points, timing-wise, relative to S phase. So now we're going to be looking at the DNA replication origins. So here we're looking at the beta globin gene cluster. This is for hemoglobin. And there are lots of genetic defects in hemoglobin, and so people study this like crazy. And somebody who was studying it saw that there were some patients that had the delta and the beta globin genes fused together, and then they, for whatever reason, I don't know, they were just like they didn't have anything else to do. They figured out where the DNA replication started there, and what they found was that there was an origin in between the delta and the beta. And that's where replication starts for this whole cluster. I guess when you want to know every single thing about it, you do that kind of stuff. Anyway, when in this patient, it's deleted, that origin. It always starts there, but now it's not there. So what this tells you, and so what will happen is in that situation, it won't make a new one. Cells won't make a new one. It'll be the normal DNA replication origin over here, just will take longer to get over there, okay? So what this says is that these DNA replication origins are fixed and they can't be replaced easily, okay? They're at set locations. Now, when they go in and sequence this and they sequence other DNA replication origins, they can't, for mammals, for like humans or any mammal, they can't find what the sequence is. They can't see it. There's nothing in common, okay? So these things are fixed, but they don't have any sequence in common, okay? We're not going to go over it for the class, but it turns out that the histones in this region have a particular methylation pattern that marks them as a chromatin. And every time it replicates, it puts a thing in, okay? So it's not sequence specific, it's based on the methylation pattern that is maintained, as the cells divide, okay? That's what determines where they go. But we don't need to know that for this class. Because it turns out that in budding yeast, they do have a unique sequence, and so that allows you to study it better. So, and this is the way that they identify those DNA replication origin things. So they take a plasmid that doesn't have a replication origin, okay? So it can't be replicated. And they just put in random pieces of yeast DNA, genomic DNA. And they're saying, oh, if I were to put in a DNA replication origin, now that plasmid will replicate, right? That's the idea. So normally this plasmid might go into a yeast, but it's just going to get lost. It can't replicate. It can't duplicate itself. But if you were to put in what they call autonomously replicating sequence, they just mean DNA replication origin, now this thing will replicate. Now you get lots of copies in the yeast, and it can be maintained among the daughter yeast. Okay? And then how do you determine whether or not the plasmid is maintained? Is that this plasmid has a histidine synthesis gene. It's required to make histidine. You're going to put it into yeast that are his minus. Okay? So like this might be the his3 gene. And you're putting it into yeast that are his3 minus. So they don't have the his3 gene. But if you put them on plates with no histidine, they can't grow. Does everybody see it? The yeast lack the ability to make histidine. Now you put them on a plate with no histidine, not growing, right? But if they can produce histidine by having this plasmid, then yeah, they can make colonies, all right? So they're just putting it in random piece of DNA, and they put it into the yeast, and the ones that can grow, clearly the plasmid could replicate, and therefore they can go in and sequence this, and they can figure out what's a sequence, okay? So they did that. Anybody have any questions about that experiment? Okay. So it turns out that there's some sequence elements. When they just sequence them, they say, oh, there's things, even though they're all throughout the whole genome, there's a lot of things that are in common, and they have these B boxes. They got A box, they got a C box. Sometimes they're lacking this B box. It turns out that this one is just a transcription factor binding site for ADF1, but it turns out they don't need that one. That one can be any transcription factor binding site. So what is the transcription factor doing? The transcription factor is coming in, and it's going to have like histone cell transferase activity. It'll be opening up the DNA. So this DNA has to open up to replicate it. So you want to have it in open conformation. So these things, whether they have this particular transcription factor binding site or a different one, doesn't make any difference. They have to be in an open conformation to be able to open up. Okay. But here we have these sequences, so you can now identify where they are in the genome because they have particular motifs. All right, so now we're going to get into, and I'll just jump ahead a little bit. These sequences like V3, V2, V1, and ACS, this A and the C, they're really short. Like this is either an A or a T, these 4 bases, a T or a C, an A or a G, 3 bases, and an A or a T. So it's pretty short. And this thing is bound by a big protein complex called the origin recognition complex. It's going to mark the origin. Okay, so that's how they know where they're going to start replication. All right, so what we're going to be looking at now is what is preventing over replication of the DNA. Basically, every time you go through S phase, DNA gets replicated once, but it doesn't get replicated more than once. So what's going to prevent it from getting replicated more than once? And so we have to get into the concept of what's called a licensing factor. So here they're going to do cell fusion experiments between a cell in S phase that's replicating its DNA, it's this slanted line, and a cell in G1 it's not. And they fuse them together, and immediately there's some factors in the S phase cell that makes the G1 cell immediately replicate its DNA. Okay? So as soon as they fuse, there's some S phase promoting factor that's going to make the G1 replicate its DNA. All right? But now if you do the same experiment with an S phase cell and a G2, so this G2 is, after it's already replicated, and you fuse them together, whatever that S phase factor is does not work on the G2 nucleus, does not replicate its DNA. Okay? So what you get from this is the concept that the cell is initiation competent. They're competent to initiate DNA replication in G1. They're not doing it, but they're competent to do it. And in G2, they're initiation incompetent. They can't do it anymore. And actually, once DNA replication starts, as we'll see, they're incompetent for starting new ones. Okay? So what we're going to be looking at is what is the license? What does G1 cells have the license? Okay? They're licensed to replicate their DNA, but they're not doing it yet, but they got the license. And now in G2, the license has been removed. Okay, so what's the license? Now, this origin recognition complex binds the origin. It's actually bound all the time. It's bound in G1, it's bound in S phase, it's bound in G2. So this can't be the license, okay, because it's there all the time. It's not changing. All right, so this is like a diagram that I made, and so this is explaining like what's happening under normal replication. So normal replication, G1 phase, the origins are licensed. They can replicate, if the right signal, but they're not replicating because they're in G1 phase. Okay, in S phase, they start replicating, but the license is removed. So the new DNA is now in red, and you get one copy. You can't restart it because there's no license. The license is, yeah. No, I can't. I'm going to get into it in the next slide. For now, all you need to know is they're licensed to do it. It's like somebody gave you a driver's license, and you can drive a car. That's a bad analogy, of course, because if you don't have a license, you can still drive a car, but you would get in trouble. So that's a bad license. But let's assume it was some kind of license where you could only do it if you had the license. Yeah, but I clearly haven't explained what a license is. That'll be next slide. Next slide. Okay, but at any rate, let's just say what happens is normal replication. So you're like, well, what's this important for? Well, why does he spend all his time talking about license? What's this license business? Well, I'll explain it to you now. What if the license is still there in S phase? You're like, that doesn't look so bad. Oh, but you don't know what's happening. What's happening is now they have the license, so they can just keep making a new one, and then when that one makes bigger, then they make a new one on that one, and then that one gets bigger, they make a new one on that one. You end up with a honeycomb pattern of DNA replication. This is the technical term, honeycomb pattern. And then, of course, it's symmetrical, so there'll be the same thing over here. Okay? Honeycomb pattern. That's unstable as hell. That thing is like, what happened to my nice DNA? It's bad news. And what'll happen is these things will just keep doing it, and they'll pop off and form minute chromosomes that are big enough that you can sort of see them. They'll be floating around. They can cause cancer. We'll talk about this when we talk about cancer. And then they can get put together by homologous recombination and get dumped into a new chromosome location where you've got thousands of copies of that thing replicated, okay? That's when it happens just one place. If this happened everywhere, it would just destabilize everything, okay? So it's super important for the cell to not allow this to happen. So they use the licensing system. So this is your answer to what license is. So the origin recognition complex binds to the origin. It's bound all the time. The licensing factors are CDC6 and CD21. This is for fission yeast, but it's sort of related to, but it's similar in all these things, but with slight variations. So in fission yeast, where it's worked out, CDC6 binds to the ORC, and then the CD21 binds to the CDC6, but it brings in a helicase. So you guys remember what a helicase is? What is a helicase? It's gonna open up the DNA, and you can't replicate DNA unless you open it up, right? To make the bases accessible. So the helicase complex is called MCM 2 through 7, but you can call it MCM. So it brings in the helicase. But this is all happening in G1 phase, and the helicase is not active yet, okay? So it's loading the helicase in G1, but the helicase doesn't get active until S phase. So what happens in S phase is you've activated the S phase cyclin CDK complex, which also activates another kinase. They're going to phosphorylate MCM, now allow it to open up the DNA, okay? So you're going to load the MCM complex in G1, but you're not going to activate it until S phase, all right? But now then the question is, why can't I just load more of it on S phase, right? Why can't I just go back in and load on more? Okay, well remember, the CDC6 and CD21 can be considered loading factors for the helicase. You need both of them working together to load the helicase onto the origin. So as soon as they get into S phase, they're both degraded. Okay? So what this is doing is it's temporarily separating the loading of the helicase from the activation of the helicase. You can never put it back on because the loading factors are missing, right? So what happens is CDC6 is degraded by SCF, ubiquitin ligase, the one I showed you before. And our lab was the first one to show that COL4, which is related to this one, degrades CDT1. So we actually were the first one to show that. So both of them are getting degraded in S phase to prevent the MCM from reloading. So these are the licensing factors. They're being regulated. They allow, so the licensing is basically the loading of the MCM complex. So here they're licensed, and then the license is removed because they can't reload it through these two licensing factors. Okay, so here's some review I wrote. This just shows that in the different organisms, these things are all working to prevent this reloading of the helicase, but they do it in slightly different ways. So this is a little bit different diagram. So here we have the ORC complex. One of them is ORC1. It binds CDT1 and CDC6, brings in the helicase. It's actually six-membered AAA ATPases. And okay, so what happens in fission yeast, once you hit S phase, as I said, CDT1 and CDC6 are degraded, so they can't load it back on. And there's also a phosphorylation of the ORC. And then in budding yeast, CDT1 is degraded, but this CDT1 home ortholog, you can call it CDT1, is exported out of the nucleus. And in fact, once the helicase finishes, it gets exported out of the nucleus too. So in budding yeast, you still don't have both of them in the nucleus where you need it, but just one of them is degraded and one is brought out of the nucleus. And then in humans, the CDT1 is degraded, but now the CDC6, instead of being degraded like it is in the yeast, is put out of the nucleus. And then the CDT1, if there's any CDT1 being made later, it gets bound by an inhibitor. And then in certain mammals, people claim that one of the ORC components is degraded, but in other mammals, it's clearly not. And so is that even true? You know, it's not even sure. We're not sure. But at any rate, so what the take-home message is that there are different ways of making sure that the helicase doesn't reload in S phase. And it's working through the two licensing factors, CDT1 and CDC6 and CDT1. Okay, and then CDK phosphorylation is going to be involved in a lot of this licensing regulation in the different species. Okay, so it turns out that you can use this system to figure out where you are in the cell cycle. All right, so so this is the Fuchi system created by Fuchi in Japan, and what he did is he took CDT1, and he and remember CDT1 in humans, CDT1 is degraded in S phase, and he fuses on an M cherry, which makes it red. Then he's going to take the geminin is only made in S phase. Geminin binds CDT1. It's only made in S phase, and he's going to add on a green. Okay? So this one gets degraded as cells go into S phase. And this one only gets made as cells go into S phase. And this one's green now, and this one's going to be red. But you can change the colors if you want. So this is the movie. Let's see if it actually works. No, I gotta open it up. All right, so the green is the geminin, and it's going to be there in S to G2, and then it gets degraded in mitosis. And the red is only going to be made in G1, and it's going to be degraded as it goes into S phase. All right. So you can see that if you look carefully, what you'll see is that it's the green cells that are the ones that are dividing. Right? They're the ones with geminin. Okay, so you see here's the green cells. And you know, one thing to keep in mind for the next lecture is when cells go into mitosis, they round up. So here, these cells round up, and then geminin's degraded, and then they hold on a second. Wait. So here, look at this cell. Okay, what? It turned into those two cells. Boom, like that. It's pretty quick. And all of them divided, right? That one divided into those two cells. And now you're in G1, and now you get the CDT1 being made, and now they're going into S phase, and then you start having the geminin being made, and then they're going to round up and then go into the next thing. So basically, from this, you can say, well, you know, you can actually look at the cells. Are the cells in G1? Yeah, they're in G1 because they're green. Sorry, red. And then you're like, are they in G2 or G2, S or G2? Yeah, they're this color. And are they in between? Then they're like orange because they're both colors, right? So you can just put this system into the thing, look at the cells, and you get a color readout of where it is in the cell cycle. So it's sort of powerful. Okay, so that's it for today.